Hello and welcome to the part 2 of our discussion of your Module 3 and Module 4 of our Biochemistry for Medical Laboratory Science. For the part 2, we will be discussing the proteins and enzymes. So a quick reminder, please keep the laboratory identification of amino acids and proteins part on your module as it will be discussed on our laboratory part. Thank you. So proteins. Okay, these are polymers of amino acids. Amino acids, these are the building blocks of your proteins. Amino acids are monomers. And when we combine these monomers, we can make up a protein, which is a polymer. Okay? Yung amino acids natin, meron siyang 20 types or 20 kinds of amino acids that make up the alphabet of the protein structure's basic structural units. So without these monomers, without these amino acids, this different type of amino acids, we won't be able to build up or to create a protein. Hence, the diversity and function. So the proteins, it has many functions in our biological functions. Okay, madami siyang ganap. And it includes the enzymatic catalysis and regulation of the proteins. Your proteins are very important molecules because they are involved in the enzymatic catalysis and regulation of proteins, number one. Number two, the transport and storage in the form of myoglobulin and hemoglobulin. In shorter terms or simpler terms, it can be called your myoglobin and hemoglobin. Next, it is the coordinated motion and mechanical support for the subcutaneous tissue and collagen synthesis. So important din si protein for the structural basis of our tissues as well, for the immune protection in the form of antibodies, the generation and transmission of nerve impulses, important si protein doon, and lastly, for the control of the growth. Okay, when we say proteins, we have classifications of proteins based on the chemical nature, the structure, shape, and solubility. Protein can be classified as simple and conjugated proteins and derived proteins. Okay, meron siyang tatlong classifications. And under your simple proteins, they are composed of only amino acid residue. Again, they are composed of only amino acid residue. On hydrolysis, these proteins yield only constituent amino acid. Then it is further divided into fibrous and globular protein. Okay, ang mga example ng ating fibrous or fibrous protein, it includes keratin, elastin, and collagen. Next, for the globular protein, it includes the albumin, globulin, glutein, and then your histones. Next classification of proteins are your conjugated proteins. They are combined with non-protein moiety. Example, yung mga nucleoprotein, phosphoprotein, lipoprotein, and metalloprotein. Okay, if you can remember, the lipoprotein, it is the combination complex of your lipid and protein. So, meron din siyang specific function on our body. Next is the derived protein. They are derivatives of degraded products of simple and conjugated proteins, and they may be primary derived and secondary derived proteins. For your primary derived, example yan sila protein, metaproteins, and coagulated proteins. And for the example of your secondary derived proteins, we have your proteases or albumoses, peptones, and your peptides. Next, for the peptide bond formation of your proteins, let us just read the definition. It is the primary amino group of nitrogen of one amino acid and primary carboxyl group or the carbonyl group of another amino acid with dehydration or the loss of water. The characteristic of the peptide bond formation includes the alpha amino and alpha carboxyl group elimination of water during the formation of bonds. And then the partial double bond character is CHNNO are coplanar. And lastly, they are less steric hindrance if transformed is favored. So ano po ba yung sinabi ni ma'am? So basically, kapag magkakaroon tayo ng peptide bond formation, again, recall lang natin si peptide bond. It is when two amino acids join together. Okay? Pag nagkaroon ng, let's say, ito si amino acid to. Ito amino acid din. So pag i-combine na natin yung dalawang amino acid, magkakaroon tayo ng peptide bond formation po yung tawag doon. Okay? And then, in the peptide bond formation, mapapansin natin na magkakaroon ng loss of water. Okay? Kasi may hydrogen na atom and then yung kabilang, let's say yung kabilang amino acid, may hydrogen atom siya. Yung kabilang amino acid, may OH group siya sa kabila. Pag nag-combine sila in the form of peptide bond formation, magkakaroon ng dehydration or the loss of water. Okay, so tingnan natin sa ating chemical structure sa baba. So pag nag-combine na yung dalawang amino acid, so mawawala na yung water natin, di ba? So ang tawag na po doon sa combination na yun, these are called your amide or amide. Okay? It is a functional group with nitrogen atom next to a carbonyl. So ang function niya, this functions as a connection of amino acid during polymerization. Again, your amide or your amide, di ba, nawala na po yung water, nagkaroon na ng loss of water or the dehydration nung nagkaroon ng peptide bond formation. Nakabuo na ng amide or your amide. It is a functional group with nitrogen atom, pansinin nyo naka-red, next to a carbonyl. Okay? This functions as a connection of amino acid during your polymerization. Okay? So going next to our functions of the proteins, we have your structural, regulatory, contractile, immunological, transport, and then your catalytic. So ito, tabular form lang siya. So basically, na-discuss na natin siya kanina. So daanan lang natin ng quick yung mga functions of the proteins. Hindi ko alam kung bakit siya nandito. For the structural function of the protein, it serves as a structural framework of the various parts of the body. Example, yung collagen that can be found on our skin, on our hair, and other body parts. Next, serves as a regulatory mechanism. It functions as hormone that regulate various physiological processes for the control growth and development, as neurotransmitters mediate responses of the nervous system. Example, yung ating mga insulin. Okay? Insulin helps the glucose to get inside our cell. Okay? Pakitandaan. Next is the contractile. It allows the shortening of the muscle cells which produces the movement. So, important din si protein when it comes to our mobility. Okay? Diyan nagtitake place or nag-action si ating myosin and actin. Next, immunological function of our protein. It aids in the responses that protect the body against foreign substances and invading pathogens in the means of your antibodies. Next function is the transport. It carries vital substances throughout the body. Pinakamahalaga po dyan si hemoglobin. Okay, siya yung nagkikerry ng oxygen in the RBC papunta sa iba't ibang tissues of our body. So without hemoglobin, hindi nakakapag-transport ng oxygen, magiging ischemic, okay? Walang oxygen na makukuha yung mga tissues of our body. So magiging mamamatay siya, mamamatay yung mga tissues on our body. Next is the catalytic function of your protein. It acts as an enzyme that regulates biochemical reactions. So your proteins, they require specific conditions to keep their shape. For example, most proteins in our bodies rely on us to keep a warm but not hot body temperature, stay hydrated, and take in enough of specific nutrients like salt. So like what we've mentioned, si proteins and other biological functions inside our body, it must be kept at a physiological state. Okay, meron lang specific pH, meron lang specific body temperature para makapag-function sila regularly. Okay po. Si proteins, katulad nga ng mga enzymes natin, meron lang dapat tayong mini-maintain ng specific pH and specific temperature para makapag-function sila optimally. Okay? If our bodies aren't able to maintain these conditions, some of our proteins may not function as well or at all. And most organisms actually produce a special protein that is called molecular chaperones that help other proteins and molecules to continue to work even if conditions are being difficult to tolerate. Agents such as your heat and urea, please do take note, cause unfolding of polypeptide chains without causing hydrolysis of peptide bonds can denature proteins. The denaturing agents destroy secondary and tertiary structures without affecting the primary structure. So mamaya babanggitin natin at i-discuss natin much further si primary, secondary, tertiary, and quaternary structures of your protein. But for now, pakitandaan po na denaturing agents can destroy the secondary and tertiary structure, but it will not affect the primary structure of the protein. Okay, so basahin lang natin yung next description. However, when a protein exposed to conditions too far outside of a range, it can tolerate that protein's shape will come undone. Okay? So tayo, parang tayo lang yan, yung ating mga proteins inside our body. Meron lang tayong, pag na-expose tayo sa isang specific condition, pwede natin siyang i-tolerate. Okay? Sobrang init sa labas. Matotolerate mo siya for a while. Okay? Meron lang certain degrees or meron lang number of hours, number of minutes na kaya nating i-tolerate yung too much heat. Just like our body, our proteins, matotolerate nila at a certain point. But, kapag ka nasobrahan na or nag-exceed na dun sa tolerable capacity ng ating protein, it will undergo the denaturing or the breaking of your protein. Magbe-break na yung mga proteins mo inside our body. So, kung natatandaan, masama po yung sobrang mataas na temperature kapag yung mataas yung lagnat. 'Di ba? Parang nagko-convulsion na yung inyong patient. Kasi nga, hindi na makapag-function regularly or optimally yung mga enzymes and proteins inside our body. Okay? So, masama po yun. So, ang tawag doon yung mga denaturing or the breaking of the protein. Masama po yun. If a denatured protein returns to its native state after the denaturing agent is removed, ang tawag naman doon, renaturation. Parang binaliktad mo lang. Okay? So, kung gagawin natin siya sa isang laboratory experiment, kunwari si protein natin, nilagyan natin ng alcohol or sinobject natin siya sa heat, yun po yun. Okay? Parang denaturing yung ginawa natin, the denaturing of your protein, which we will perform on our laboratory part. Okay? Kung kahit laboratory experiment at home to, please be prepared. Gagawin po natin siyang experiment sa laboratory. Okay, so going back to the main topic, denaturing, kapag sinubject mo si protein into a breaking agents. Okay, so pag binalik mo naman siya, pabalik dun sa kanyang natural state, ang tawag naman dun, renaturation. So kasi, naturally, our proteins are folded. But once you subject it into a breaking agent, it will unfold. Okay? So these are the types of protein structures. So meron tayong 4 types. We have your primary, secondary, tertiary, and then your quaternary. Okay po. So discuss natin siya sa susunod na slide. So for the primary structure of a protein gives the details of the amino acid sequence of a protein. By convention, the sequence of information is written from left to right, starting with the N-terminal amino acid and ends in the C-terminal. Please do not forget. Left to right, N, and ends with the C-terminal. The primary structure of a protein will determine all other levels of structural organization of a protein. So for the hierarchy of the structure of your proteins, it will start from the primary up until the quaternary. So pinaka-basic talaga dyan si primary. Next, the primary structure of a protein will determine all the other levels. Next, the primary structure is stabilized by peptide bonds, which are covalent in nature. Please do not forget, the most important element of primary structure is the sequence of amino acid residues. Okay, so highlight po natin siya. How will I highlight this one? Highlighter. Highlighter. Okay, I cannot stress this enough. The most important element of primary structure is the sequence of your amino acid residues. Bakit po? Discuss natin siya later on. Next, si isoelectric pH, the net charge is 0 and the protein does not migrate to anode or cathode. Okay? And then the primary structure of protein will provide information regarding the proteins. If it's 3-dimensional structure, function of the protein, the cellular location, and then the evolution of the protein. Next, in general, proteins contain at least 40 amino acid residues. Polypeptides smaller than are simply called your peptides. Then 40 residues is the minimum for a polypeptide chain to fold into a discrete and stable shape that allows it to carry out a particular function. Next, your secondary structure. The secondary structure includes various types of local co-informations in which the atoms of the side chains are not involved. Secondary structures are formed by a regular repeating pattern of hydrogen bond between backbone atoms. Thus, secondary structures are stabilized mainly by your hydrogen bonds. Okay? And then the two most important secondary structure in the protein, please do not forget, is the alpha helix and then the beta plates. Due to the formation of hydrogen bonds between peptide bonds, two types, we have yung coiled natin and then yung sheets or pleats. Okay? Ang difference po nila sa ating structure is that your coils or your helixes, it is brought about by intrachain of hydrogen bonding. Intrachain. Pag sheets, okay? Pag sheets, it is brought by interchain of hydrogen bonding. So ito yung itsura ni secondary structure. Okay? It has two different types, yung alpha yung tsaka yung beta. Si primary as is residue lang talaga. Okay? Next is for your tertiary structure. It refers to the unique three-dimensional structure of the protein, including the geometric relationship between distant segments of the primary structure and the relationship of the side chain group with respect to each other in three-dimensional space. Pagka meron ka nang nakitang three-dimensional structure of the protein, automatic tertiary structure na yung pinag-uusapan natin doon. Okay? And we have two general classes of proteins based on the tertiary structure. Dalawa lang siya, your fibrous and then your globular protein. Your fibrous proteins, highlight lang natin, which serve mainly structural roles, have simple repeating elements of secondary structure. Okay, yun lang siya. Si globular naman have more complicated tertiary structures, often containing several types of secondary structure in the same polypeptide chain. The first globular protein structure to be determined by X-ray diffraction method was your myoglobin. Okay, so yung fibrous or fibrous protein natin, parang linear lang siya in shape. Yung globular natin, para siyang naka-coil na naka-circular shape yung ating globular protein. At ang classic example niyan, which is yung pinakaunang protein structure nga under globular protein that was recognized and was determined is your myoglobin. And then these are the types of interactions that stabilize your tertiary structure. We have your hydrophobic interaction, the electrostatic, the hydrogen bond, and then your covalent bond. And lastly, the protein folding is assisted by the molecular chaperones. Okay? Madami talagang naitutulong si molecular chaperones dito, lalo na sa protein folding. Protein folding of a polypeptide chain does not occur by a random search. Rather, protein folding appears to be a cooperative sequential process in which formation of the first few structural elements assists in the alignment of subsequent structural features. Also, the folding pattern and the final conformation of a protein are dependent upon its primary structure. The two types of proteins, your enzymes and molecular chaperones, are required for the proper folding of the proteins in the cells. And lastly, the quaternary structure. Basahin lang natin yung mahabang description and then discuss natin siya dun sa susunod na slide. Meron tayong picture doon. Most proteins, particularly those with molecular masses about your 100 kD, consist of more than one polypeptide chain. Each polypeptide component is called a subunit. Okay, subunits in a protein complex may be identical or quite different. A multi-subunit protein is called multimer or multimer. A multimer with just a few subunits is called oligomer. And their identical units are called protomers. A large number of oligometric proteins contain two or more subunit protomers referred to as dimers and tetramers respectively. Monomeric proteins consist of a single polypeptide chain. Dimeric proteins contain two polypeptide chains. And then your quaternary structure is exhibited only by proteins containing more than one polypeptide chain. Please do not forget your quaternary structure is exhibited only by proteins containing more than one polypeptide chain. These chains or subunits are held together by non-covalent interactions such as hydrophobic interactions, electrostatic interactions, and hydrogen bonds, as well as interpolypeptide disulfide bonds. Hydrophobic interactions are the principal forces holding subunits together, although electrostatic forces may contribute to the proper alignment of the subunits. And lastly, quaternary structure designates the characteristic manner in which the individual folded polypeptide fit each other in the native conformation of an oligomeric protein. So ano ba yung mga sinabi mo, ma'am? Ito po siya. Example natin si hemoglobin. Okay? It has 4 separate polypeptide subunits. Okay, medyo complex kasi yung pagkakabuo o yung structural uh, characteristic of a hemoglobin. Thus, it falls under the quaternary structure. Okay, hindi po siya tertiary kasi it's not under the fibrous protein or globular protein, hindi din siya secondary, hindi din siya lalong primary. It is under your quaternary because marami siyang polypeptide chains. Okay? Most proteins, particularly those with molecular masses above 100 kD, consist of more than one polypeptide chain. Thus, your hemoglobin. Okay? So, in a nutshell, in a simpler term, at i-summarize na natin siya, your protein structures, when we say primary, sequence of residues lang siya. Okay? Paulit-ulit. Pag secondary, localized folding lang. Meron na ka nang nakitang folding sa secondary. Pag tertiary, complete folding pattern. And then, pag quaternary naman, meron ka nang napansin or na-observe na interaction ng mga tinatawag po nating subunits. Okay po? And then, in remembering or studying your specific type of diseases, it is important to know the molecular level on which these biomolecules has something to do with a specific type of disease. Okay? So katulad nung nabanggit natin, sickle cell disease, yes, meron siyang problema sa protein. But what type of problem? It has something to do with the protein. Okay? Meron bang missing? Meron bang sobra? Meron bang na doble? Meron bang na triple dun sa structure? Okay? When we say sickle cell disease, meron siyang mutation doon sa specific amino acid. Okay? So sabi nga po natin, mahalaga yung sequence ng isang amino acid, mahalaga yung sequence ng isang DNA doon sa kanyang structure. Okay? Kasi nagkaroon lang ng isang mutation, nagkaroon lang ng isang pagkakamali doon sa isang amino acid, yung isang normal individual, nagkaroon ng mutation sa kanyang isang amino acid, napalitan yung glutamine ng valine, nagkaroon na siya ng sickle cell disease. So that mutation affected the individual, hence yung kanyang cell imbis na biconcave in shape siya, nagkaroon na siya ng sickle cell shape red blood cell. So hindi siya maganda, hindi siya nakakapag-carry ng yung hemoglobin niya, hindi nagpa-function regularly, hindi siya makapag-carry ng oxygen into the other tissues of the body. Okay, so ito po yung significance ng pag-aaral natin ng ating mga diseases when it comes to the molecular level. Okay? Next, enzymes are types of your proteins like what we've mentioned. Enzymes are specialized protein catalysts that accelerate chemical reactions. Okay? Walang sabi-sabi, walang isip-isip pag sinabing enzymes. Pag sinabing enzymes, accelerate chemical reaction. It speeds up the chemical reaction. Yun lang. Enzymes agad ang sagot doon. Okay, in biological systems, so about 105 to 1,014 times faster than uncatalyzed reactions. So yung enzymes, tumutulong sila para mag-speed up yung mga reactions, chemical reactions happening inside our body. So imagine kasi kung mabagal, 'di ba? Sobrang matagal yung growth, yung digestion, yung metabolism, ang tagal. So mas maganda na meron po tayong regular functioning enzymes working inside our body. And at the end of this, neither used up but are regenerated, recycled, and utilized. Next, ibasahin lang natin yung mga definition of terms. The APO enzyme, it is a protein part of an enzyme, main enzyme, catalytically active, cannot increase reaction rate. Next, the cofactor is a small organic or inorganic molecules either loosely or tightly bound to your APO enzyme that participates in overall catalytic activity. And then meron kang three cofactors. Okay? We have your coenzymes, wag kakalimutan, your prosthetic group, and then your metal ions. Okay? Ang enzymes natin, meron lang siyang two parts. Madami lang definition, pero basically, ang enzymatic activity in our body, meron lang siyang dalawang components na kailangan. Yun po si enzyme and substrate. Okay? Important lang po talaga dyan si enzyme and substrate. Okay? Itong si active site, part lang siya nung inyong enzyme. Bakit ba nakaturo siya sa substrate? Dito lang siya sa enzyme. Okay? Yun lang po yun. And then your substrate is the molecule acted upon by the enzyme to form your product. So, the goal is pag nagkaroon ka na ng combination, pag nag-join na si enzyme and substrate, makaproduce ng product to speed up the catalytic activity on your body. So, these are the characteristics of your enzyme. Sobrang haba. Basahin na lang natin sila ng mabilis. Ano bang mahalaga dito? Ito lang, mostly proteins in nature, although a small number of RNA-based biological catalysts are called your ribozymes, okay? And have identified as proteins. They are made up of chains of amino acids joined by your peptide bond. The catalytic activity depends on the integrity of the native protein, and with denaturation, their catalytic activity is lost. Ano pa bang mahalaga dito? Highly specific for the reactants and substrates they act on. Also, nakalimutan ko, balikan ko lang. Okay? Highly specific po si enzyme and then si substrate. Highly specific in a way na hindi siya makikipagkabit, hindi siya makikipag-join kung hindi yun yung substrate niya. Si substrate, hindi naman siya makikipag-join kay enzyme kung hindi yun yung enzyme niya. Gets po ba? Highly specific sila to one another. Okay, si enzyme gusto niya, meron siyang specific substrate. Si substrate, doon lang siya sa specific enzyme makikipag-join. Gets po ba? And then, ito, balikan natin sa active site of the enzyme. It is the machinery involved in the catalyzing reaction. So, pag may mga nakita kayo na mga terms na may mga A's sa dulo, ito po sila. Kadalasan, mga enzymes po yan. So, basahin lang natin yung mga categories of our enzyme classification. Number 1, your oxidoreductase. It functions as the transfer of electrons, results in a change in oxidation state. Mga example natin dyan, yung mga dehydrogenase. Next, yung mga transferase natin, it functions as a transfer of functional group from one molecule to another. Example natin yung mga phosphorylase and your kinase. Next, your hydrolase. It is the breakdown of covalent bond using your water. So, mapapansin naman natin sa ating prefix and then dun sa suffix. Ano siya? Breakdown of covalent bond using water. Hydrolase. Okay? Example, yung mga proteases natin and phosphatase. Next type is your ligase. It is the breakdown of covalent bond without water or oxidation. So, yun yung difference nila ni hydrolase. Ang example natin dyan, yung mga decarboxylase. Next, your isomerase. It is the rearrangement of bonds within a molecules. Example natin yung mga mutase. And lastly, your ligases. It is the formation of covalent bond between two large molecules. So, for our enzymatic activity, we have two models of substrate-enzyme bonding. Pakitandaan, pakiaral po ng maigi. Number 1 is the lock and key model. The substrate binds to a site whose shape completely complements its own. Okay? Parang lock and key. Diba? Ano naman, evident naman, obvious naman sa ating ginamit na term. Lock and key. Kung hindi yun yung lock, hindi din yung key, hindi po sila magkakaroon ng enzymatic activity. Highly specific sila to one another. It does not take into account 3D flexibility of protein. So si substrate, kakabit lang siya doon sa kanyang enzyme kapag sila ay specific to one another. Next, for the induced fit model, not rigid, undergoes a slight conformational change on binding substrate induced by multiple hydrogen bonds forming a complementary shape or a flexible pocket that approximates the shape of the substance. So, kay lock and key, highly specific sila. Kay induced fit, pwedeng may mag-adjust. Okay, nag-a-adjust po yung inyong substrate. Ay, nag-a-adjust yung inyong enzyme. Okay, kunwari hindi sila fit. Pagpasok ni substrate papunta dun sa kanyang enzyme, si enzyme nag-adjust. Nag-fit siya para kay substrate. Okay po. And then ito po yung mga factors that affects our enzymatic activity. Pakitandaan si active site. Enzymatic catalysis depends upon the activity of amino acid side chain assembled in the active center, then the temperature and the pH. Okay, for our enzymes to work optimally, meron dapat tayong sinusunod na certain temperature and pH sa ating katawan. Okay? And lastly, the concentration and type of the substrate. Enzymes require an optimum temperature and pH for their action. The temperature or pH which compound shows its maximum activity is called optimum temperature or optimum pH respectively. Typically, enzyme activities are accelerated with increasing temperature. As enzymes are functional in cells, the feasible conditions for nearly all enzymes are in temperatures that are moderate. At higher temperatures, given a specific point, there is a drastic decrease in the activity with the denaturation of the enzymes. So, ang mechanism nito, si enzymes, meron lang tayong physiological pH at which nagkakaroon siya ng optimal activity. So, pag tumaas yung temperature sa loob ng ating katawan, nagsi-speed up din siya. So, tumataas din yung activity ni enzyme hanggang sa na-reach na yung kanyang certain point, magdi-decrease na siya eventually. Pababa na ng pababa yung kanyang activity. So, hanggang sa hindi na nagpa-function talaga yung inyong mga enzyme. Okay? Oo, mag-accelerate siya sa kasabay ng pagtaas ng temperature nyo. Pero meron lang certain point na pag na-reach ni pH, yung na-reach na ni enzyme yun, magdi-decrease na siya eventually. And then this is the kinetics of enzyme catalyzed reaction. So, hindi naman po siya dapat kabisaduhin at mag-focus. Ang importante lang dito, alam natin yung factors that affect the enzymatic activity, which is the temperature, pH, and then the salt concentration. At saka po itong picture sa baba. Okay po. So, let us just discuss the general mechanism of your enzyme action. Okay, so for your general mechanism of enzyme action, pansinin po muna natin yung picture dito. Okay, yung ibig sabihin niyan enzyme. Okay, yung S substrate. So kapag nag-combine na si enzyme and then si substrate, makakabuo sila ng tinatawag nating enzyme substrate complex. Iyon yung ES. Okay? And then after some time, pagka nagkaroon ka na ng enzyme substrate complex, it will produce a product and then yung enzyme mo na ulit. Okay? Again, enzyme substrate. Pag nag-combine na sila, makakabuo ka na ng enzyme substrate complex. And then eventually, it will form a product and then another enzyme na gagawa na naman siya ng another activity. So, katulad po ng nasa picture natin dito. Okay? When a specific substrate combines with your enzyme, it will form an enzyme substrate complex that will eventually become a product and then will form a new enzyme again. Okay? So, kung bakit po may K1, K2, K negative 1, K negative 2, para po yan dito sa ating formula for the Michaelis-Menten kinetics, which we will not discuss further kasi medyo complex na siya. Sobrang haba na siya. In a way na dapat magkaroon tayo ng separate discussion for this. Okay? Pakiaral lang po kung ano yung ibig sabihin ni Michaelis-Menten kinetics kasi it involves this formula. But basically, hindi na tayo mag-dwell on dyan. Okay? Ang importante, alam natin yung general mechanism of our enzymatic action, which involves two components, which is your enzyme and then your substrate, then that will eventually produce an enzyme substrate complex, and then it will produce a product and then an enzyme again. So, kung meron tayong ganung mechanism, meron tayong syempreng hadlang, di ba? Parang mga palabas sa TV, kung may bida, meron kang inhibitors or may hadlang. These are the inhibitors of your enzymes. So, meron tayong 3 types. So, in a normal setting, meron kang enzyme, meron kang substrate, and then tuloy-tuloy ang kanilang function. So, meron tayong tinatawag na competitive inhibitor, meron tayong tinatawag na uncompetitive inhibitor, and meron din tayong tinatawag na non-competitive inhibitor. So, discuss lang muna natin ano po si competitive inhibitor. Competitive inhibitor diminishes the rate of catalysis by reducing the proportion of enzyme molecule bound to a substrate. Again, it diminishes the rate of catalysis by reducing the proportion of enzyme molecules bound to a substrate. Okay, kita naman po dito sa ating competitive inhibitor. Next, for your uncompetitive inhibition, it's a substrate-dependent inhibition that the inhibitor binds only to the enzyme substrate complex. The uncompetitive inhibitor binding site is created only when the enzyme binds in the substrate. Okay, ito po yun. Meron kang uncompetitive inhibitor once na nag-combine na or nag-join na si substrate and then your enzyme. Okay, uncompetitive kasi nandun lang siya sa may active site. Pansinin? 'Di ba? And then lastly, your non-competitive inhibition or your non-competitive inhibitor, the inhibitor and substrate can bind simultaneously to an enzyme molecule at different binding sites. Okay? Dito, sa part ng enzyme, pwedeng kumabit dito si non-competitive inhibitor, pwede din dito, and then hindi na nag-fit fully si substrate dito. Okay, nag-fit siya partially but not fully. Hindi na sila nakapag-function regularly. Dahil nga meron kang non-competitive inhibitor. And then your allosteric enzymes, it binds activators or inhibitors at sites other than the active site. And then plots versus generate the sigmoidal curves. The allosteric activators cause the enzyme to bind substrate more readily shifting the kinetic curve to the left, thereby decreasing the apparent Km. And then your allosteric inhibitors cause the enzyme to bind substrate less readily, shifting to the curve to the right, increasing the apparent Km. And these are my references. Thank you for listening.